Hi friends, welcome to Life Science. This channel is exclusively for covering various topics in life sciences. The topic is development of brain in frog. Before going to the topic, please subscribe my channel, press the bell icon for notifications, like, share among your friends and comment. Development of brain in frog occurs after gastrulation. During gastrulation, there are morphogenetic movement take place. As a result of this, finally the gastrula contain three germ layers: ectoderm, endoderm and mesoderm. Then brain and nervous system derived from the embryonic ectoderm. In the gastrula, the presumptive material for nervous system lies on the mid-dorsal line as a plate called neural plate. Soon, the edges of the neural plate become thickened and rise above as neural folds. The neural folds increase their elevation and bend towards one another until their edges meet and fuse and a tube is formed called neural tube. The anterior part of neural tube is distinguished as encephalon or brain, which develop into various parts of the brain through thickening, thinning, evagination and invagination. The primary embryonic brain of frog has three main subdivisions called prosencephalon, mesencephalon and rhombencephalon. The cavities of three primary divisions are known as prosocele, mesocele and rhombocele. We can see about prosencephalon or the forebrain. The prosencephalon become further subdivided into two regions, the telencephalon and the diencephalon. At the posterior limit of prosencephalon, the brain bends ventrally around the anterior end of the notochord to form cranial flexure, which remains as a prominent feature of brain of vertebrates. The area of cranial flexure thickens to develop the tuberculum posterior that marks the posterior limit of the forebrain ventrally. The most anterior division of the forebrain is the telencephalon with its original cavity, the telocele. The anterior limit of telencephalon is the lamina terminalis, which will separate the future cerebral hemispheres by a longitudinal groove. Actually, the lamina terminalis represents the anterior fused neuroporal area. The telencephalon is the embryonic cerebrum. Its cavity expands laterally to give rise to the right and left lateral ventricles and the surrounding thick-walled cerebral hemisphere at about the 12 mm stage. The two telencephalic vesicles are partially constricted off from each other but remain connected by way of tubular foramen of Monro, which open into the common third ventricle. The third ventricle overlaps and connects the telocele and the diocele. The roof of the cerebral lobes thickens to give rise to cortex or pallium, and the floor and side of which form the corpora striata. The olfactory lobes arises as a pair of evaginations from the anteroventral part of telencephalon. Subsequently, they became fused medially. The nerves originating from the olfactory lobes innervate the nasal epithelium or olfactory placard. The ventricle enclosed by the olfactory lobe is called olfactocele. The next part is diencephalon or thalam-encephalon or between brain. The structural derivatives of diencephalon are posterior commissure, epiphyseal recess and epiphysis, pineal gland, then anterior choroid plexus and habenular ganglion and commissure. The paraphysis. Infundibulum, then infundibulum ingrown and from hypophysis or the pituitary gland of the adult. The hypophysis become the anterior part of the pituitary gland. During metamorphosis, the individual lobes of the pituitary gland differ both in gross morphology and in finer structure. Between the infundibulum and the tuberculum posterior is a secondary and posteriorly directed pocket known as the mammillary recess. The other structures seen in diencephalon are optic chiasma, optic recess. In front of the optic recess, there appears a ventral thickening called torus transversus. Then optic vesicles are there, optic cup will form. then retina, pigmented layer and optic stalk, optic chiasma, all these can be seen during the development of eye also. The stalk will develop around an inverted groove that is called choroid fissure, which will contain within the groove accessory nerves and blood vessels which feed the retina. The next part is mesencephalon or midbrain. Midbrain is in between prosencephalon and the posterior rhombencephalon. Then these having found principally within the paired ventrolateral thickening of the wall and floor on either side of the tuberculum posterior. They are known as crura cerebri. Then optic lobes or corpora bigemina is present. Then valvulae cerebelli, the original cavity of midbrain, connect to the rhombocele or the fourth ventricle with the third ventricle, which become narrow and is known as the aqueduct of Sylvius. Next region is called rhombencephalon or the hindbrain. This portion of the brain is clearly marked off from the mesencephalon by a transverse constriction in the roof of the brain at the posterior limit of the dorsal thickening. It is not clearly divided further. There appears a slight transverse thickening in the roof of the rhombencephalon, which correspond to the metencephalon of higher forms and develop into the small cerebellum. Then posterior choroid plexus is present. The ventral and ventrolateral wall of the rhombencephalon are known as medulla oblongata, from which arise the cranial nerves 5th to 10th inclusive. The wall becomes thickened by fibers which form numerous pathways from brain and cord. The rhombocele or cavity of the hindbrain is known as the fourth ventricle, which communicate posteriorly with the central canal of the spinal cord and anteriorly with the aqueduct of Sylvius of the mesencephalon. Here shows the divisions of the developmental stages of brain in frog. The first figure shows telencephalon, diencephalon, metencephalon, myelencephalon, and the extension is called the spinal cord. Then here are the cross sections of the brain in various stages, and the final diagram shows all the parts of the brain with the ventricles. From this diagram and explanation, I hope you could understand how the development of brain in frog take place. Dear friends, thank you for watching. Don't forget to leave a like and please subscribe my channel.